In the next series of videos, we'll be demonstrating the activities associated with the pulmonary respiration practical. In particular, we'll be looking at how to use and calibrate the spirometer pod. We'll also be looking at how to measure lung volume and flow, how to measure the forced expiratory volume in 1 second, and also the forced vital capacity. And we'll have a look at the effect of airway obstruction, as well as airway restriction on those measures. And lastly, we'll use a different machine called a vitalograph in order to also measure forced expiratory volume in 1 second and forced vital capacity in order to compare those with the readings that we got from the spirometry pod. Now, to set up the spirometry pod, let's first of all have a look at the equipment that we'll be using. First of all, we'll need a power lab, a bacterial filter, a marker to write our name on the bacterial filter, a nose clip, some alcohol wipes just in case you need them, and a spirometry flow head connected to the spirometry pod. First of all, take the sterile bacterial filter from its bag. The round end is the end that will insert into the spirometry flow head, which we'll see in just a sec, while the oval piece that you can see here is the mouthpiece that you will breathe through during the activities shown in these videos. During a full practical, we'll generally get you to write your name on your bacterial filter so that you don't get it mixed up with anyone else's. Once that's done, insert the round end of the filter into the spirometry flow head. There's a blue rubber adapter down one end of the flow head that you should insert the bacterial filter into. Now that we're all set up and ready to go, the first thing that we need to do is to calibrate the spirometry pod. To do this, click on the Zero Inputs button just underneath the lab chart window in your CuraCloud notes. This will zero the pod. Then pick up the flow head and stand up, put your nose clip on, and click Start on the lab chart window to start the recording. You can then start breathing into the mouthpiece, and your ventilation will now be recorded in the lab chart window. The notes ask you to record about 1 minute's worth of data. Once you have at least 1 minute's worth of data, you can click Stop. Then resize the screen so you can see the data that you've just collected. Now you should notice that the volume is changing over time. In the example shown here, you can see that there's a slight downward slope of the data. The reason for this is that the air that you breathe in is dry and generally cool. When you breathe that air in, it then becomes humidified, so water vapor is added to that volume. And the air is also heated to body temperature, and so now the air is warmer and therefore takes up a larger volume than the air did that you inspired. The result is that the volume that you exhale is slightly larger than the volume that you inhale. And so we need to make a slight correction for this, and that's what this calibration is doing. As described in the notes, to make this calibration, select at least 10 seconds worth of data. In the example shown here, we've selected 41.5 seconds worth of data, which is fine. Scroll down and you'll see the calibration window, and you can see that the spirometry is currently uncalibrated. So just click the Calibrate button. And you'll notice that the calibration then occurs, and you can see that the data which was sloping downwards is now horizontal. Now there's one more calibration that we need to do just over on the next page. So if you click over to the next page, you'll see that there's a Flowhead Spiropod calibration section. Written on a piece of masking tape on your flow head will be a calibration value. To get this value, we used a calibration syringe to pump a known volume of air, in this case 3 litres, through the flow head. We then measured what the flow head calculated this volume to be. So in this example shown in the video, when we pumped 3 litres of air across the flow head, we actually measured 3.14 litres. We've set up the tables in your CuraCloud notes to do a correction for that flow head, and so if you type in the value that was measured for your particular flow head, so in this case 3.14 litres, then all the other values will be corrected for using that value. Your spirometry pod and flow head should now be all set up and ready to go on with the rest of the practical.